you can go.
So good morning, everyone. It's my pleasure to have Yann Bourgeois today. So Yann is an evolutionary biologist. He's currently a lecturer at Portsmouth University in the UK. So he's interested in population genomics of fast adaptation, the dynamics of transposable elements, and the application of population genetic tools to conservation. So in terms of background, he did his PhD at Toulouse, dealing with the genetic basis of color variation in an endemic bird species from Réunion Island. Then he did a postdoc in Ebert's lab in Zurich to work on host-parasite interactions. And then he spent 3 years at New York University, Abu Dhabi, where he worked on multiple models, including toads and anoles. So Yann is an expert in population and genomics of adaptation and has a substantial track record given his career stage. And nowadays he's launching multiple projects in population genomics of transposable elements in a variety of species and also in conservation genomics. So today is gonna talk more particularly about teas. So Yann, it's your turn now. Floor is yours. Thank you for accepting the invitation.
Oh, well, thank you, Marielle, for this lovely introduction. Hello, everybody, even if I cannot really see you. Nice to meet you all. I'm going to share my screen now. So is it working?
Perfect.
Okay. So again, thank you for welcoming me here to invite me to give a seminar here in Montpellier. So today I'm going to talk about the population dynamics of transposable elements. My perspective is more the one I'm coming from a population genetics background. So I'm going to talk about how we can maybe use population genetics, or at least I hope, to understand better how transposable elements behave and how their diversity is shaped in species and within genomes. So usually when you start talking about transposable elements, you start with this observation that vertebrate genomes and genomes in general vary a lot in size. So here this is a figure that comes from this paper from Capusta et al. In PNAS from 2017. And what they did was that they looked at all the results that were obtained in terms of estimate for genome size in many different vertebrates. So you can see that, well, there is quite a lot of variation, but it depends on the clades. For example, you can see that in birds, genomes are relatively short. They're about 1.2 gigabases, and there is not much variation. Whereas, for example, in the case of rodents, you have a lot of variability. So with genomes that can go from 1.5 gigabases to even possibly 6 gigabases. And of course, what underlies this is that the genomes are dynamics when it comes to genome size. So here on the right, you have a graphic of phylogeny that shows you the relationships between different species of vertebrates. And what they did is that they used a method to estimate the ancestral genome size. And from there, they tried to estimate the amount of genome loss, gene loss and gene gain, DNA loss and DNA gain, in the genomes. And what's interesting is that most of the gains that you observe come from these elements in genomes that are called transposable elements. So one of the main questions that you can ask is what is driving this variation in genome size? So one hypothesis that was proposed by Michael Lynch and collaborators at some point was that the genome size is directly correlated with effective population sizes. So the idea here is that when you have large populations, the effects of selection against new DNA fragments, for example, or against a large genome size is stronger, and genomes tend to be more compact. There is also the idea that life history traits can play a role. When you have smaller genomes, you also have smaller cells, for example. So they can have a role in, for example, the rate, the generation time, or like the, for example, in the case of birds and bats, it has been proposed that the short genomes that they have could be linked to adaptation to flight. Can be also, of course, due to the fact that genomes have intrinsic properties in terms of gene content, gene density, and, for example, epigenetic variation that can lead that can facilitate, promote, or impair the gain of DNA or loss of DNA. So, as I said, a lot of these events of gains, a lot of these size gains, they seem to come from transposable elements. So I'm going to shortly introduce what they are. They are often described as genome parasites. Transposable elements, they are DNA fragments in the genome that are able to replicate and insert somewhere else in the genome. Usually, they are divided into two main categories, which consist in retrotransposons, which use a RNA intermediate, and DNA transposons. So the way retrotransposons work, you have a progenitor that contains all the elements that are needed for its replication. So this progenitor is going to be transcripted. So you have RNA. This RNA itself will be translated, so it will produce all the proteins such as transposases that are necessary for the element to then be reverse transcripted and inserted somewhere else in the genome. So it's a copy and paste mechanism. To be perfectly clear, some of these elements, they are actually non-autonomous. So you can divide into several subcategories these transposons. You have, for example, the lines, you have the structure here, or the long terminal repeats retrotransposons. They differ in the way they insert. You also have the signs, which those ones that are non-autonomous. So it means that they need lines, for example, to provide the necessary proteins, the necessary enzymes to be able to transpose. On the other hand, you also have DNA transposons. Those ones, they use a cut and paste mechanism. So you have a progenitor, which is going to be transcripted, translated. So the translation will lead to the production of a transposase that will excise the progenitor and remove it. So in the end, you have an empty site, and this progenitor is going to be inserted somewhere else in the genome. So to keep it short, you basically have retrotransposons, which behave following a copy and paste transposition mechanism, and you have DNA transposons, which behave as cut and paste. And as I mentioned, genome sizes are strongly correlated with the amount of Ts. So if you look at the proportion of Ts in genomes, you will see that they are made up of various percentages of different T families, so signs, lines, LTR, RTs, or DNA transposons. The relative proportion of those different families is not necessarily the same across multiple genomes. It looks like there are specific dynamics in each for each family. But what's really interesting is to see that there is indeed a correlation between genome size and T content, usually. So why am I interested by population genetics to study transposable elements? Well, you can see that here on this small graph, genome size is impacted by the amount of transposable elements that I insert, for example. And this depends directly on the transposition rate, so how frequently a given transposon is going to copy and paste or cut and paste. This transposition rate can be controlled by the host, so this rate is going to fluctuate over time, depending on whether the host manages to control transposable element transcription. There are also mechanisms of preferential insertion. Some transposable elements tend to favor some specific sites in the genome, or they recognize specific DNA sequences that they prefer, or they are attracted by regions of open chromatin. So, but what's ultimately going to have an impact on genome size is the fixation rate. So it's not enough that you have a lot of transposons inserting in one genome. At some point, the resulting genome needs to spread in a population of and these mechanisms, they are controlled by well, population genetics mechanisms such as selection, genetic drift. So we can actually look at all these different processes, which are transposition rate, fixation rate, through the lens of population genetics. Why is it useful? In my opinion, with these, we often consider them in the light of comparative genomics. So all these different graphics that I showed you before, you can see that we look at, for example, genome composition, genome length, at relatively large time scales, long time scales. So we are looking at comparative genomics. We compare genome contents of different species. The host history is not necessarily taken into account. And this is where population genetics can actually be interesting, because you can make predictions about the relative frequency of different elements in the genome depending on the host history. So far, we know that there are some transposable elements that can be adaptive. So there are a lot of examples where transposable elements, because they insert in front of a gene, they disrupt its function. So either they knock out the gene, or because some transposable elements contain promoter sequences, promoter domains, they can sometimes also lead to changes in the expression of the gene. So this has been shown, for example, in industrial melanism of moth in the UK, for example, where the black moth that are cryptic in a black background, they actually carry a transposable, actually multiple transposable elements just in the promoter region of the gene that encodes, that is responsible for melanism. So we have like a few anecdotal evidence that sometimes transposable elements can be recruited by the host, domesticated, so they provide an advantage in some specific environmental conditions. We also have evidence that a lot of them behave in a way that's consistent with neutral expectations from a population genetics perspective. And actually, since we often consider them as genomic parasites because they insert anywhere in the genome, they often disrupt the function of genes, they are deleterious, and they are often found under purifying selection. But we don't necessarily know exactly what's going on in terms of the relative abundance of these different categories. How many TEs are actually adaptive? Are they always adaptive, or is it really context dependent? What's the fraction of TEs that are neutral? So basically, what we are trying to do with population genetics is to understand, not only for TEs actually, what is the distribution of fitness effects of new mutations or new variants. So in the case of TE, the same question applies. We try to understand what are the fractions of TEs that will be adaptive under purifying selection or neutral, and what is the strength of selection against all four TEs. So I think that population genetics can provide us a way out to go a bit beyond a descriptive aspects of TEs biology by examining the abundance and diversity of transposable elements in populations. So we don't look at one single genome, but we compare diversity across multiple genomes of the same species. And we can compare these patterns with markers that we assume are closer to neutrality, for example, such as single nucleotide polymorphisms that you can find, for example, in intergenic regions. So today, my plan consists mostly in showing you a few examples of how I think the population genetics can be applied to the study of TEs. So what can we infer, for example, from genome-wide variation in TE frequencies? I will spend some time explaining why the age of TEs matter, because as I was mentioning on this slide, we have some transposition rate is something that can be controlled by the host, and it's something that actually is important to take into account in population genetic studies of TEs. I'm also going to detail a bit why it's interesting to look at where TEs fall in the genome. So basically contrasting not only looking at TE frequencies, but looking at how these average TE frequencies vary in different genomic regions. Then I will try to illustrate how these different aspects can be used to study TE variation and how they can provide us with some information using a model that's the green arrow. So this is the self-promotion part, let's say, of my talk, where I talk about my own work. And then I will conclude with a few ideas and perspectives that I hope maybe could be pursued further. But basically the goal is to give you an idea of what I think could be interesting ways of estimating the distribution of fitness effects for TEs and not just their average effect on the host. So before we start, I also just want to give a very brief description of the way we detect transposable elements in genomes. So what I'm going to do for the rest of the talk is that I'm going to talk about transposable elements that are polymorphic in populations. So we are going to look at all the sites in the genome that carry a transposable element, and we are going to examine those TEs that are found in one genome but not the other, for example. So the way we detect them, at least from short reads data, which at the moment are the bulk of data that are available, let's say, for population geneticists. We basically take the reads, the short reads from Illumina sequencing, and we map them back on a reference genome. So the reads that map back perfectly, they are not clipped, for example, that's fine. But the reads that are interesting for us are the ones that are split. For example, let's say that here we have transposable elements in a specific individual that we just resequenced. this transposable element is not found in the reference genome. So what's going to happen is that you have those reads in black, they map to the reference, and at some point, there is part of the sequence that actually maps on the transposable element. If you have paired end data, so basically you sequence DNA fragments in two directions. So that's what this shows here. So you have like the black parts correspond to the sequence fragments, and in the middle there is a fragment that is not sequenced. So when you map them back, normally there should be about 500 basis pairs, let's say, between the two fragments that are sequenced, the two parts that are sequenced. But it can happen also that you have one, for example, the forward read maps back on the reference genome, that's fine, but the second one will map on a transposable element that is found in the reference. And this transposable element can be found very far away or even on a different chromosome. So most of the methods that exist at the moment, what they do to detect transposable elements that are not in the reference but are found in one specific accession or individual that you are resequencing, is that they take all those reads that show discrepancies. And what they do is that they take the part that does not map back on the reference genome and try to map them on a consensus reference for different TE families. So this way you can identify the genomic regions where you have a potential TE insertion. It works also on the other way around. So if you have a TE in the reference genome, but it is not found in an accession that you are sequencing, you will see a gap. And that can also be used to call the TEs that are found in the reference, but are not found in the genome that you are resequencing. So here, at the moment, one of the, let's say, best tools that exist is called MELT. What's interesting with MELT is that not only can it detect TEs that are present or absent in the genome, but it can also tries to genotype them. So when you are working with diploid species that are not, for example, two inbred, which is the case of many animal species, you can determine whether a TE insertion is heterozygous or homozygous. The main issue with this method, for example, is that it can be very slow. The field is moving really fast, so I wanted also to highlight the existence of this resource. So there is a web link here. So if you are interested, I can also provide the presentation. The link is there. So it's called TE Hub, and it lists all the tools that are currently generated to address, basically to call TEs in genomes. But yeah, this is basically how we detect polymorphic TEs in genomes at the moment. The main difficulty, of course, is that if you are interested in population genetics, you need to be sure that you are genotyping correctly your individuals, especially if you expect them to be heterozygous. And this is actually surprisingly hard. So here I'm highlighting an interesting paper that has been published recently in Nucleic Acids Research, where so the lead author is Clément Goubert. And what they did is that they looked at the accuracy of calling, for example, using MELT or other approaches, and they compared them with the genotypes obtained when you genotype through PCR, the transposable elements. What's a bit worrisome is that when you look at the accuracy, it's not too bad when you are calling, for example, when you are calling for absence of TEs, homozygous absence. Heterozygous TEs, not necessarily that bad using MELT, for example, but when you call, for example, the genotypes that are supposed to be homozygous for the TE, you see that the accuracy drops a lot. Fortunately, they managed to develop a method that regenotypes the output from MELT, and it manages to increase quite a lot the accuracy. That can be an issue because when you look at population genetics, you want to have good estimates of allele frequencies, and this is something that needs to be taken into account, I think, in future studies that will examine allele frequencies. The main issue is that this algorithm is nice, but it works only for allele elements in human genomes at the moment, but hopefully it will be expanded. I just want also to note that this is a general issue. So even if we are studying, for example, single nucleotide polymorphisms, the colors also can have a hard time depending on the species that you are studying. I'm thinking of maize, for example. Okay, so now we can go to the population genetics part. So what can we infer? What can we expect in terms of TE frequencies, depending on different mechanisms? So we can use the information inferred from single nucleotide polymorphisms to estimate what we call an allele frequency spectrum. So we look at how many, when you sequence, for example, 10 individuals, you have 20 alleles, and you just count how many loci have one derived allele, 2 derived alleles, 3 derived alleles. So in the end, you produce this sort of graphic where you count the number of derived alleles in the case of SNPs, or the number of T inserting in a population of genomes. And basically, the shape of the allele frequency spectrum tells you a lot of things. So the shape will be different depending on the demography. So here we have an example where we looked at signs, which are very short transposable elements. And we compared two populations of lizards. One of those populations we know by using population genetics approaches, we know that one population has been quite large, 1 million individuals for a long time, and the other population is bottlenecked. So it went through a sharp reduction in effective population sizes over the recent times. And what you can see is that the shape, when you look at different T families from the science different T clades from the science family, you can see that in the large stable population, you have a lot of transposable elements that are quite rare. So you have this peak here, let's say, for low T counts. Then you have this U shape, which is quite classic. What's interesting is that when you look at the bottleneck population, you see that you have a lot of you have more alleles, you have more Ts that are found at intermediate frequencies. So this is an effect that you expect if Ts are behaving neutrally, let's say. So you can use this and compare. What's interesting also is that you can use these allele frequency spectra. You can also see them shaped by the effects of selection. So here I'm taking the same two populations, but this time I'm looking at another family of Ts, long terminal repeats. Those ones are supposed to have a stronger effect on the host because they are longer and they are likely to have deleterious effects. And you can see actually that they show a lot. You have a lot of elements that are found at low frequencies in population, in both the large stable population and the bottleneck population. And you don't see much of difference in the spectra. So this is something that you expect in the case of purifying selection, for example, acting on transposable elements. Something I should mention is that the spectra that we see here for the signs, they are quite similar to the spectra that you would see if you were looking at markers such as SNPs in intergenic regions that we assume should be neutral or closer to neutrality. But there is another further difficulty with transposable elements, which is that here, for example, we see that we have a lot of rare elements in population. But this can be due either to the fact that you are counter-selected, so you never reach high frequencies in populations. Another possible factor is that those elements are just quite young. And this could be due, for example, to recent transposition. So something that has been proposed for transposable elements in different species is that they don't necessarily have a transposition rate that is constant. So there is this hypothesis that you have some elements that display bursts of transposition. So this is shown on the left here. So you could have a transposition rate that is relatively constant or doesn't change much over time, in which case you could consider transposable elements the same way you would consider, for example, single nucleotide polymorphisms with a relatively constant mutation rate over time. And then you could assume that the spectra, the allele frequency spectra, when you compare them between SNPs and Ts, well, basically the deviations between them should be due to factors such as selection, for example, assuming that demographic factors affect all markers in the same way, which of course can be discussed. But if transposition rates are non-constant over time, for example, if you have a very recent burst of transposition, you expect that most of the Ts that you observe in the genomes, they inserted recently. So they didn't have enough time to reach high frequencies in the genome, in which case you expect also this excess of young transposable elements. So you would expect this sort of allele frequency spectrum. So you would expect an excess of singletons or like rare variants compared, for example, to single nucleotide polymorphisms. On the other hand, if transposable elements were active a long time ago, but are not active anymore today, what you have is mostly a population of transposable elements that are old, so they have had the time to drift to higher frequencies in the genome. So in the case of an ancient burst of transposition, you would observe a spectrum that's much more flat compared to the single nucleotide polymorphisms. So this is what I'm showing here on the right figure. Does it work? So how can we correct for that? So there are several approaches that have been suggested. For example, we can estimate the age distribution of transposable elements, even from a single genome. You possibly saw this sort of graphs already. When you read a paper describing a new genome, a new reference, people often use an algorithm called repeat masker, repeat modeler, to estimate the fraction of the genome that is made up of different families of transposable elements. So here, for example, we are looking at the green anole. I'm going to tell more about this species later. And you can see, so the different bars correspond to different types of elements, so signs or lines. And what you can see here on the x-axis is an estimate of the substitution level. So what you do is that you create a consensus for each of those different subfamilies, these different clades, and you look at how divergent the Ts that you observe along the genome are compared to this reference. And this gives you an estimate of the dynamics of Ts. So when you see a lot of elements that do not display any strong divergence from the consensus, it suggests that those elements amplified recently, transposed recently. And when you have a lot of elements that are very divergent from the consensus, it suggests that they are much older. So it gives you an idea of the activity of different types of transposons over time. So this is something we could possibly include, for example, in models when we are trying to estimate basically how T's age could impact the shape of the frequency spectrum. So there are two different papers I want to highlight there that I found quite interesting. The first one was a paper by Justin Blumenstiel. It was published in 2014. And basically what they did is that they had access to the transposable element sequence itself. And by comparing for the same family of Ts, let's say, or for the same T clade, the number of substitutions, pairwise substitutions, they can estimate the age distribution of all those elements. And what they do is that they create a genealogy of all these elements. And based on that, they can estimate when transposable elements have been more active. And then they can inject the age of transposable elements in a model, in a Bayesian model to be precise, that will take into account both the age of Ts and the allele frequency spectrum to estimate the average effect of selection against Ts. The average selective coefficient against Ts. So this method, it requires access to the transposable element sequence, but it's quite an elegant method that basically tries to correct for this issue of recent transposition, for example, in overestimating the effects of purifying selection on transposable elements. A more recent approach that was, last time I checked, actually, it was in bioRxiv, but I think it was recently accepted in Genome Biology and Evolution. It was described by Horvath and Rossibara, 2021. And this time, it's probably more appropriate for people like me, for example, who don't necessarily have access to the T sequence itself, but have access to the flanking sequence, so the flanking genomic regions. So what they do here is that it's always the same idea. You try to correct the frequency spectrum of Ts by taking into account the age distribution of said Ts. This time, instead of estimating the age distribution of Ts from the T sequence itself, they use the flanking regions. So what they do is that they use this algorithm called GIVA, which is based, I think, on estimating the length of tracks that are identical by distance. And based on this length, you can estimate the age of different haplotypes. So they estimate the age of the Ts based on that. And then they create an allele frequency spectrum that is corrected. So what they do is that they extract SNPs, single nucleotide polymorphisms, that match the age distribution of the transposable elements. And by comparing the allele frequency spectra this way, they can estimate the selective coefficient, because again, it's the same idea. You can estimate the selective coefficient by comparing near neutral markers to the selected ones and look at how the two spectra are, well, how the T spectrum is shifted compared to this neutral expectation. So this is just to summarize a bit, well, what we can do with, in terms of population genetics based on the allele frequency spectra of Ts and comparing them with derived SNP frequencies, for example. So if you assume, for example, that Ts transpose at relatively constant rates, and they are scattered through the genome in the same way than SNPs, you would expect that the allele frequency spectra should be really similar between Ts and SNPs, for example. And in that case, the shape of the spectra is mostly, if the Ts are neutral, the shape of the spectra is mostly shaped by changes in population sizes and demographic changes over time. To simplification, this is what you would expect. If Ts are under purifying selection overall, they have deleterious effect, or if you have recent burst of transposition, you expect an excess of rare and young Ts. And if you have an ancient burst of transposition, for example, you would expect an excess of Ts at higher frequencies. And as I said, there are now a few methods that are available to take into account the fact that Ts do not necessarily have constant transposition rates to still be able to estimate deviations between neutral expectations and selection. Another aspect that's interesting to consider, and this draws from all the recent literature about what is called linked selection, consists in looking at where Ts are in the genome. And from this perspective, an interesting statistics is the recombination rate. So why is it important? It's mostly because, for example, Ts, transposable elements, well, one hypothesis for why they are so deleterious to the host in some cases is that they can engage in what we call ectopic recombination. The idea is that if you have a lot of transposable elements that are quite long, so they share the same sequence or similar sequences, and they are found a bit everywhere in the genome, where they fall in regions of high recombination, these regions engage more frequently in recombination, which means also that those transposable elements are more likely to trigger ectopic recombination, which means recombination between non-homologous sequences in the genome. This is extremely deleterious. This can lead to aberrant meiosis and then ultimately very low fitness. There's also the fact that, at least in the green anole, so the organism that I'm going to describe now, in the green anole, you also have correlations between the rate of recombination, gene abundance, and regions of open chromatin. But another, at last, aspect that's interesting about recombination rate is that it can be used as a predictor for the strength intensity of the effects of linked selection. So what is that? So here I'm just giving an example where you have, let's say, two populations. So just focus on population 1 here. Let's say that you have a deleterious mutation that occurs. Well, in regions of low recombination, you will have a very long haplotype that's removed from the population, and this haplotype is associated with the deleterious mutation. And basically what happens in the end is that you expect that regions of low recombination display less genetic diversity, less nucleotide diversity, for example, for SNPs, than regions of high recombination. So what you expect also is that in these regions, you will observe a trend where allele frequencies tend to be biased, so basically you will have a lot of elements that you will have a lot of stochasticity due to higher genetic drift. Or at least that's equivalent to regions of locally lower effective population sizes. So you expect less diversity for neutral markers. On the other hand, in regions of high recombination, you expect more diversity because you have more independence between adjacent loci. So you don't have these effects of reduced diversity due to linked selection. It's not only about purifying selection, also the same effect would be observed if you have a positively selected locus. On the other hand, because you have a lot of correlations between recombination rates, chromatin content, and gene content, it can also be that there is also the fact that some transposable elements tend to be more attracted to either regions of low recombination or high recombination. So what you will have here are two mechanisms. Linked selection will act on T frequency once they insert. And the differences of Ts for preferential insertion will lead to changes in the density of Ts that insert in the given region. So this is a summary of what I just told you. So you can, again, look at the frequency of transposable elements, but you can look also at the location and the genomic context. So you can look at the markers that are linked to specific Ts to make predictions about, well, and you have different expectations in terms of T frequencies, but also the diversity around Ts, depending on whether the T is involved, for example, in positive selection. So you would expect in that case to have very long haplotypes flanking the Ts, or depending on whether the T is under the direct effect of purifying selection or under the indirect effects of selection, for example, linked selection. So an example of application of all those different concepts in an actual model, that's something we tried to do with my former advisor, Stéphane Bossino at New York University, Abu Dhabi, so 2 years ago. We worked on the green anole, which is this species, this little guy that lives in Northern America. What's interesting is that it occupies a very broad range of environments, from the southern Florida to actually more temperate zones, let's say in Carolinas and Midwest. So it could. What's interesting with the green anole is that it displays a really high diversity of transposable elements. So you have, you can see here this graphic that shows you in different colors the relative abundance of different families in the reference genome. And you see that the 4 main families of Ts are found there. So you can find the signs, you find also the lines or non-long terminal repeats recruit transposons, also long terminal repeats recruit transposons, or DNA transposons. So what we did at the time is that we called for different populations of the green anole polymorphic Ts. And then we looked at their frequencies in different populations and for across the different families and clades. So what's interesting in the green anole again is that it shows really strong genetic structure. So you can find 5 different genetic clades. You have 2 clades that are found in more temperate regions here, so in purple and orange. And you have 3 clades that are found in Florida, and they are all clearly distinct in terms of genetic diversity. So you have 2 different representations here. So we have those 5 different clades. What's interesting is that the 2 northern clades here, if you look at the changes in effective population size over time, you can see here on these different curves, the populations from Florida, they apparently have been quite stable with effective population sizes of about 2 million individuals for the last million years. Whereas in the last 100,000 years ago, the populations from northern latitudes show an evidence for bottlenecks. So this is consistent also, again, with the first graphic I showed you about T frequencies here in those 2 populations. What's also interesting is that you can clearly see the impact of what is called linked selection on genetic diversity. So the first panel here shows you an estimate of the recombination rate along the 6 chromosomes of the green anole. What you can see at the bottom is a statistics that measure diversity along those chromosomes, and you can see that they match quite well. So where you have high recombination rates, you tend to see more diversity at SNPs, and where you have low recombination rates, you see much less diversity. It also leads to faster drift, which means that when you compare genetic divergence, genetic differentiation between the different populations, you see that it tends to be higher in regions of low recognition because you have faster drift happening in these regions, and it tends to lead to a better discrimination of the markers that you sequence in these regions. So there are many factors, as I mentioned before. So we have like the possibility that Ts are impacted by changes in transposition rate. TEs also might be more attracted by some regions in the genome than others. So what we did is that we created a quite simple model to understand the patterns of diversity that we observed in the genome. So what we did is that we simulated using this simulator called SLIM3, which is becoming quite popular in population genetics. And what we did was that we tried to generate simulations that were relatively similar to what we expected in terms of diversity in the green anole. So we focused on the population in Florida that's stable, that has been stable over time. We generated fragments of 4 megabases. And along this genome, we made the recombination rate vary. And we generated simulations where transposable elements could burst a long time ago for 100,000 years, or they displayed constant transposition rates, and they displayed either no preference for some regions, like the regions of high recombination rates, or they actually did display preference for the said regions. So we generated the expectation for 3 main statistics: the average frequency of polymorphic TEs, so how many TEs do we have that are polymorphic in genomic windows of 1 megabases, how many polymorphic insertions we find in the same regions, and how many insertions we observe that are fixed in the same regions in the population. So they are informative, these statistics, because they describe in the end different categories of variants of TEs, either the ones that have reached fixation, the ones that are still in the process of reaching fixation, and the average frequencies of the ones that are not fixed yet. And all these different fractions, well, the relative abundance of those different categories will depend on the demography and selection. So this is the complicated graph that we generated in the end. So those are like the simulations, the result of the simulations, and what we expect under different combinations of parameters in the different scenarios. And we can observe that we have radically different observations in terms of, for example, the abundance of polymorphic insertions in regions of high recombination or low recombination, depending on whether the TEs were neutral or near neutral, so with low selection, weak selection against them, or under, in the case TEs had strong selection against them, especially in regions of high recombination. So just to give you an example, this is an example where we can clearly see that the transposable elements display patterns of diversity that are consistent with purifying selection against them. We can see, for example, that the frequency of gypsy elements tend to be much lower in regions of high recombination. There are also way less polymorphic insertions in regions of high recombination. And we also see way less fixed insertions in this region. So this is a classic case that's consistent with our simulations, where we simulate markers, TEs, that are under purifying selection. So this would be this example here. So you look at the red boxes, and this is what you expect. If you have strong purifying selection against TEs, and that this selection is stronger even in regions of high recombination, for example, due to ectopic recombination, you expect indeed a negative relationship. On the other hand, when you look at signs, they tend to display patterns of that are consistent with neutrality. So you see that there are more, the average frequency is higher in regions of high recombination. And this is probably due to the fact that in regions of low recombination, the effects of link selection tends to reduce diversity. You also observe more polymorphic insertions in regions of high recombination. What's interesting also is that you observe more fixed insertions in these regions of high recombination. This is not necessarily something that we expected, actually, under a purely neutral model. So if we just look, we would expect that actually, assuming signs are neutral and under the impact of link selection, we would expect actually them to accumulate more, to fix more frequently in regions of low recombination, which are here in the middle, which was not the case. So to explain that, we need to include in the model preferential insertion in regions of high recombination to explain what we observe. What's interesting also is that if you include even more information about the element, for example, in the case of lines, we can also provide an estimate of their length. And we can see that the longer these elements are, and the less frequent they become in regions of high recombination, which again is consistent with what we expect in terms, for example, of ectopic recombination. So if you have long elements, they are more likely to be involved in this deleterious mechanism, and therefore become less frequently, more strongly selected against in regions of high recombination. And this is something that we observe. So it's interesting to incorporate information about Ts themselves, as I was trying to illustrate with the example from the Bloom and Steele study that I mentioned before. It's limited at the moment with short read sequencing, but the recent advances in long reads might actually help us creating more integrated models. So ultimately, I'm not going to go in details there, but we generated possible mechanisms that explain for each of the different T-clade mechanisms, or combinations of mechanisms that could explain their diversity. So for example, mostly linked selection and preferential insertion, or purely linked selection, or a combination of purifying selection against Ts in regions of high recombination and demographic effects. So where can we go next? So this is a pilot study. The thing is that we just generate qualitative expectations depending on different combinations of mechanisms, such as purifying selection, preferential insertion, rates of transposition over time. But what would be very interesting in the end would consist in using more information and build models that will allow us to estimate a selective coefficient, for example, and to estimate even the distribution of selective coefficients against or for T's insertions. So an idea that I hope to get funded at some point would consist in in estimating the genealogies near T's. So we have methods nowadays that can estimate the relatedness between genomes for different non-recombining blocks along the genome, and using these genealogies, you can make you can identify the genomic regions which are under the effects of direct positive selection or purifying selection, under the indirect effects of selection, or that are by looking at all these those genealogies, you can reconstruct the past history of species. So what would be interesting, in my opinion, would be to contrast the genealogies near transposable elements and contrast them with genealogies in the rest of the genome. The idea then would be also to fit, for example, linear models using these local genealogies and extracting information about allele age to contrast regions that contain Ts or nearby flanking Ts with the rest of the genome. Another idea that I like that was developed in 2009 by Venner, Fechot, and Guillemot is the idea that we can maybe and we can think of genomes as let's say niches for transposable elements, and we can apply maybe some concept of community ecology to understand how transposable elements reach equilibrium in genomes. So in the case of community ecology, you have different hypotheses, for example, niche partitioning or divergence. The idea would be to apply the same sort of concepts on transposable elements. So for example, instead of butterflies here, you would have different T families. So are those families just occupying the space that's available, that's it? Or are they actually, is there any pattern of niche divergence where Ts actually adapt their strategy depending on the competition that they have with other Ts? It could be something quite interesting to investigate. I'm currently trying to discuss with a few people and trying to see where we can maybe try to combine those population genetics approach with this kind of framework that's drawn from community ecology. If you're interested, you can look at the papers from Venner, Fechot, and Guillemot, which displays quite a few interesting ideas. The main difference with classical community ecology maybe would be that in our case, we would have competition between the different ecosystems. So that would need to be included in the model. And this is possibly where we can introduce concepts from population genetics. And once we have another idea would be to improve the modeling process. So what we can do is that we now have the simulators that are needed to generate expectations in terms of T diversity, depending on the patterns of preferential insertion, the strength of background selection or linked selection, the fact that they have non-constant transposition rates. Maybe we could include this idea of niche competition to simulate different T families at once. and see how they insert. And the idea would be from there to estimate the selective coefficients against TDs, and estimate possible distributions of these fitness effects on the host. We can also use these models to take into account what I was mentioning at the beginning, the fact that we have still issues with possible genotyping errors. So we can, hopefully the goal, but it's a long-term one, would be to obtain some kind of inclusive approach that takes into account possible biases in the generation of data and the different biological processes underlying TS diversity to ultimately draw an expectation, let's say, for selective coefficients. So to do that, there is a framework that's quite interesting, which is a framework of either approximate Bayesian computation or approaches such as machine learning and deep learning. To put it simply, and because we don't have that much time also, we can just take, we can create simulations using all these different parameters I was mentioning, and we contrast them with the observed data. And once we have, we do this contrast, we can try to estimate which set of simulations is the best at estimating the combination of statistics that we observe in the data, and we can then draw the distributions for the most likely sets of parameters, the most likely combination of parameters that would explain the data that we observe. So it's always Bayesian in the end. It's the probability of the model given the data. So to do that, I propose that to study 4 different species. So I'm interested in vertebrates, for example, and we could examine TEs at different stages of their amplification in 4 different models: the green anole, humans, and mouse. So it's interesting because they differ in the TE content, so they don't necessarily have the same dynamics. They don't have the same genome size. It would be also interesting to include the Drosophila in there. And there are different expectations that we can draw because they don't have the same genome size. For example, we don't necessarily expect the same distribution of fitness effects. In compact genomes, a T insertion is more likely, for example, to insert in a region that has a function, biologically speaking, from an evolutionary perspective. So maybe we would have a higher variance in selective effects with a lot of Ts that are strongly deleterious and a few Ts that are strongly positively selected. Whereas in species with longer genomes, you might expect a lot of Ts to just fall in regions that are already non-functional, in which case they have very limited effects on the fitness and selective coefficients that are close to 0. It's actually quite interesting to note, for example, that in Drosophila in experiments, they suggest that 50% of visible phenotypic changes might be due to T insertions, while in humans it's probably less than 1%. So thank you for your attention. I hope I was not too long. I might have been, sorry. So I want to to acknowledge my former advisor, Stefan Boissino, and for the study on anoles, Robert Ruggiero, who is now who is now a professor in the United States, and Imtiaz Aryani, who was an undergraduate at the time, but who helped a lot in terms of discussion, and who is now a PhD student in California. Thank you for your attention, and sorry for the time it took, I realize.
Yeah, thank you very much, Yann. Yep. So we have time for some questions. I think there are 2 questions online. First by Queen B. Yep. So if you want to Queen to ask your question aloud, please.
Hello. I can see them in the chat, actually.
Yes.
So, yeah.
Okay, then
I'm just gonna repeat them maybe.
Yeah,
for
probably. Yep.
Mm-hmm. So a question from Queen Bui, I think. Approaches to detect polymorphic Ts. Did you try REPET, a well-known tool developed by URGI to annotate Ts? Do you have an idea about the difference between REPET and TE-Hub? Sorry, just to be clear, TE-Hub, it's a resource to list different possible tools, from what I understand. It's not a tool by itself, or maybe it is, but I haven't seen that. I didn't try REPET. We used MELT directly, which was supposed to be state of the art at the time. It would be indeed something to do. So in general, if I'm going to carry on with these studies, I need to well, me or the postdoc who's actually going to do it, we need to benchmark different methods, definitely. So this is what I wanted to highlight also with these different methods that are now used for genotyping. It's clear that we're not fully there yet when it comes to genotyping Ts. I think it's still easier, for example, in inbred species, because at least you expect them to be mostly homozygous, so it helps a bit. For heterozygous species, it's still a bit difficult. How do we test the linked selection with Ts? Ts could play a role in the gene selection during domestication. Yeah, so it's funny that you mentioned that, because I actually would like to study Ts from a domestication perspective. So it's with Muriel, actually, we would like to look at them. To test linked selection with Ts, so that's not easy, to be honest, because linked selection, the idea is that it will reduce diversity over a quite long region. So I'm not sure if your question is about can we actually disentangle, so can we differentiate between Ts under direct selection and Ts that are just near a site that is itself in the selection. That will depend, for example, on the sample size. So the more samples you have and the more you can refine the window in which you have a strong signal of selection, for example. But ultimately, and this is something also I want to emphasize, we cannot just use population genetics alone. We need at some point to include functional studies. And this is where the field should move towards, the direction towards which the field should move. But it's not, of course, easy for all species. But Ts do play a role in selection during domestication. You have a few examples where Ts, for example, insert near a gene that's involved in color changes of the fruit, for example. So it's the case in grapefruits, for example. It's the case in date palm. It was the case also in maize.
Then probably, Yann, so yep. to come to this first question and bringing that to a more general level, then you were saying of the differences between when, to approach the question of whether you know the sequence or not of the Ts, no, for instance. And then, but then coming back to the initial question is that as it happens in all approaches that are heavily bioinformatic based, so how volatile are the results depending on the development of new technologies or of new algorithms? Because this is actually, I think, one of the questions in there. And then how volatile they are and how dependent are they then, for instance, on the generation of novel sequences. Let's say that now we can do with PacBio things that are longer, and then that allows you. So how volatile are the results? And what is the need then of being constantly generating new data or of reanalyzing old data?
So I would say that at the moment, from what I can see, but of course it can be discussed, but from what I can see, when you have like transposable elements that are quite frequent, usually they're quite robust. You are not. So for example, presence absence, I think we are doing quite fine. When it comes to genotyping, this is where we have a lot of progress to make. Then there is also the issue of identifying the exact T family or the T clade. And that's where actually long reads are going to help us a lot, because the approaches I was mentioning, you just sequence at most maybe 100 basis pairs of each side of the T. And for the ones that are truncated, for example, that's something that's, well, that can be enough, let's say. But for the ones that are quite long, you don't get enough information about what's happening inside the T. So you can try to estimate that. You can try to estimate an age. It's still a source of information, but that's, I think, indeed where the long reads are gonna probably change a lot of things. In terms of annotation, maybe not the fact that there is a TE there, but what kind of TE is it? That's also something that's gonna probably change a bit.
Muriel, probably then she will have.
Yeah, I have a question regarding what you said at the end of your talk. So you mentioned that there is a relationship between fitness effect and the genome size. So the fact that the genome is larger, you may have a lower deleterious effect due to T.
It's more a hypothesis at the moment, but yeah.
Oh, okay. Yeah, so okay, regarding that, I was wondering, like, in the end, it's like a vicious circle. The more you have TE, the more they can accumulate in the genome. So is that why you think we have like extreme cases of T accumulation like in maize? Do you think that that's an explanation?
So that's actually a mechanism that has been proposed for some plant genome, for example, that are very rich in Ts. And there is this idea of runaway effect, where when you reach a given genome size, a given threshold, you start having you don't have enough regions, let's say, that are functional in a way. That Ts on average, will have an effect, let's say. So the idea is that the probability for a T to insert in a region that's functional becomes less and less. And at some point, you have this kind of runaway mechanism by which the genome can increase to really large sizes. But then you also have the fact that for species that have, for example, large population sizes, you don't necessarily expect, you expect like even Ts that insert in regions that are not necessarily, or let's say Ts that have very low effects on fitness in very large populations, they are still going to be counter-selected. So you expect also this effect to be important in controlling the genome size. There are other aspects that are interesting in that case. For example, the idea of what happens to asexual species. So there was a recent paper about deloid rotifers. So they reproduce asexually, and they are trying to test, for example, what you would expect in species that do not recombine anymore is that you have less purification against Ts, and you have Muller's ratchet also. So the fact is that you can accumulate genetic load. So even deleterious Ts can accumulate, especially if the population is small. And you would expect, again, genome sizes to increase. On the other hand, because you are asexual, you don't have these mechanisms, the genetic load can become so high that you also select for mechanisms that will excise or control for the amount of Ts. And this is actually what they tend to observe in deloid rotifers. So they see that it looks like the Ts are more active, but also they tend to be more repressed. So there are clear signatures of selection against on genes, positive selection on genes that control T proliferation, for example. So you see, it's always this sort of dynamics and thresholds dynamics. That's quite interesting to study.
So probably we have.
Thank you.
Probably we have the time for a question. My last question, it's already half past, by Julie Clément. So so probably you can ask your question aloud.
Yeah. I can repeat
Julie,
the question.
if you're there, you can manifest yourself.
Yes.
Great.
Hello. Hello. I will ask my question. So, yes, we know that transposable element can also promote or impair recombination locally. So, do you think it's an important parameter to take into account in your models? Because this role can also modify the link selection locally?
That's a good question. So I know that there are people at ERD who are working on actually trying to refine the recombination maps of rice. So basically to predict recombination landscapes based on T content, because of what you say, actually, the fact that when you have more Ts, for example, you increase divergence between regions that normally would recombine, you can decrease recombination rates. That's something that I think would be interesting to include in the evolution, for example, of sex chromosomes, like, you know, when recombination stops between the regions that are associated with the sex determining genes. I think that at the moment I want to create models that are a bit simpler, and then we can increase in complexity, but that's definitely something to take into account. But maybe for situations where we know that this mechanism might play an important role, for example, in species isolation or in the case of selection. But yeah, definitely.
Yes, it can also have a role in maybe hybrid sterility or some stuff like that. Yeah.
No, yeah, I do agree. So technically, it should be doable. I mean, knowing the simulator I'm using, you can actually, change the recombination rate depending on whether an element insert in the region or not. So that could be something to include.
Thank you very much.
Thank you.
Good. I think that probably because it's already 35, so 25 to 1. So probably we could close in here. So thanks a lot, Yann, for the talk, and thanks all for your time. And then I would like then to remind you all that next Friday the seminar will be at 4 p.m. We are hosting Arthur Weiss from Toronto. So next Friday we'll meet then at 4 p.m. for the next seminar of the Ecologie-Evolution Montpellier. So thank you very much, and have a good weekend, everybody, and see you on Friday.
Thank you very much.
Bye.
Bye.